Using a microscope can be very helpful. This year we're going to be using microscopes quite frequently in life science and in chemistry. Though this tool has been influential in both biology and in chemistry, it is only useful when we know its proper function, and that involves knowing the different parts of the microscope itself. As we look at the different parts of a microscope, it helps us to understand its use and its functionality. Let's begin looking at the beginning part of this, which is the eyepiece. This eyepiece is attached to what was called the body tube. The body tube is then attached to, in this case, a rotational cup that allows for it to rotate. Not all microscopes have this function. Commonly, the tube doesn't have any magnification itself. It is simply a conduit that allows for us to see down to the objective lenses. These objective lenses, as you can see, come in many different sizes. That allows for different magnification. Above the objective lenses, we have a revolving piece that allows them to rotate from side to side, allowing us to change the magnification as we look at specific specimens on a slide. As we look at specimens, we generally put them on this black counter, which is known as the stage. The stage also has these clips, known as stage clips. These clips hold the slide in place so that as we look at one particular part of a slide, we're able to find that more easily. This also allows us to show someone else something that we have observed within a specimen or a specific location within a substance. It is important to be able to communicate to others what you have seen, and so these stage clips allow for us to show and point out different features. Along those lines, inside the eyepiece, one of the things that you'll see is that there's usually an arrow, and as you look through that, it helps to point out specific areas that you have located in your slide or specimen that you have attached to the stage clips. Continuing, we have underneath the stage the diaphragm. This opens and closes, allowing more or less light to come through. The diaphragm is important because as we may want less light for a specific specimen to see more or less contrast, being able to change that amount of light is really important. The light itself comes down here from the light source, usually a lamp of some sort with different types of lumens, meaning brighter or lighter based upon the type of bulb that's inside. Over on the right here, we have different adjustments. This bigger adjustment is known as the coarse adjustment. As you're first trying to get the focus of the specimen, we generally use the coarse adjustment to get it into focus with a relative precision or clarity. As we want to look closer or in more detail, we want more clarity, and this fine adjustment allows for us to get that much crisper of an image, allowing us to see maybe details that we wouldn't have seen had we not used the fine adjustment. Both are very important, but generally the coarse adjustment is used first. In the end, we have two main components that we'll hold on to as we move a microscope. This is going to be true for any type of microscope. In this case, we have a picture of a compound microscope. So down here we have the base. The base is one of the strongest points of this microscope. We would put a hand underneath this, and our other hand goes on here, the arm. The arm is the part of the microscope that is going to be metal and hold a rigid frame. This allows for the microscope to be carried carefully from one location to the next, as we'll do from shelf to table and back from the table to the shelf. If you have any other questions, bring them to class and we'll discuss this more. Remember, you will be having a quiz on this specific information. If you need to replay the video, please do so.